Okay, so yesterday we were talking about that populations, we're starting to look at how we can see if a population is evolving. And one way to do that is to look at the frequencies of the alleles in the population. And so if a population, if you recall, is not evolving, the frequencies of your big R and little r in the population will stay the same from generation to generation. And so then we started talking about yesterday ways in which this this doesn't happen, in which the frequencies can change. So remember that there were those 5 things that need to be true about a population in order for the population not to evolve. And so one of those things was having a large population. And so what I was starting to talk about yesterday when we talked about this thing called genetic drift, it has to do with small populations and how they can cause changes if one of 2 different things can happen. And so there's the bottleneck and the founder effect. So I'm going to give an example here of this. And so yesterday, I think I asked you guys to figure out the frequency, figure out the calculation of how they figured that out. Did I do this yesterday? Okay. And so, so remember that there were 10 flowers, so there's 20 total alleles, and 14 of them are the red gene, and so there was 14 out of 20 to give us 0.7, and then we can figure out Q as well. And so then what happens in genetic drift is if something, like if it's the bottleneck effect, the bottleneck effect means that something catastrophic happens and randomly kills off part of the population. And if it's a small population, this is a really big deal. And that's what this picture is trying to show you. So if we look at this and the individuals that, let's say a fire comes through the forest and kills off these guys here. And so these ones with the boxes, that's supposed to be the ones that are left. So when it says only 5 out of the 10 plants leave offspring, those are the survivors. Those survivors don't necessarily have the same frequency of genes that the original population did. And so out of these 5, if we look here, we have 2 red genes, 3, 4, 5 red genes out of a total of 10 genes now, because there's only 5 individuals, so 5 out of 10. Now our frequency of the red gene is 0.5 instead of 0.7. It's gone down. So it's not representative of the original population. So then they lead their offspring, and then to just hit the point home here that let's say another catastrophic event occurs and these guys are killed off and these 2 are left, and so therefore all the offspring will be red, and so we have a big change in the diversity of the population. And this happens more often with small populations. It's not as catastrophic usually if there's a large population. All right, that's called the bottleneck effect. Why it's called the bottleneck effect, this is an example here. Here's an actual bottle, all right? So you can see the neck of the bottle gets skinnier. And so there's marbles, this is an analogy, there's marbles in these bottles. The marbles represent the different individuals and the diversity of the individuals. And so what happens in the bottleneck effect, like this fire randomly kills off a portion of the population, and it's random which ones survive, just like it would be random if you turn this bottle over and shake it. You get a few marbles that come out and you have a few surviving, and that surviving population isn't representative of the original. All right, and then this last picture here, did I talk about the seals? No. This last picture here is with these elephant seals in California. At one point they were almost hunted to extinction, so there were very few left, and so they've been breeding them to try and bring back the population. It's illegal to hunt them now, but the seals that were left had a reduced variation, so this new population has less variation than originally the seal population did. All right, which then of course the less variation there is can hinder its adaptation and be able to have a variety of traits, individuals that have a variety of traits to help it to survive in their environment. So that's a real life bottleneck effect. And the founder effect is the same principle, except instead of a bunch of the organisms dying off, it just means that you have a population let's say of 1,000 individuals, and let's say 10 of them separate from the population and go over here and found, like founders of a new area, so they go and inhabit a new area, those 10 individuals may not have all the diversity in traits that those 1,000 individuals did, and then they reproduce, and so this population will have different diversity and different traits, and their frequency of big R and little r will be different than the original population. So that's genetic drift, and so that's how a small population can change the P and Q. So the result of genetic drift is a reduction of genetic variation within a given population, and that can increase the differences between populations of the same species. Another exception to the Hardy-Weinberg is that can change the frequency of the big R and the little r is this thing called heterozygote advantage. Individuals are heterozygous at certain locus or locations, which give them a survival advantage. So, and this can result in non-random mating and survival, which then can change the frequency of the big R and the little r. So we're going to use sickle cell disease as an example here. We've talked about sickle cell disease. Sickle cell disease, remember, is a recessive disease, and that it's caused by a point mutation in the DNA. Throw my thing away. A point mutation in the DNA. And so what does that mean? One base is changed, so it substitutes one base for another. Well, what that does is change the mRNA and then changes one codon of the mRNA, which then changes one amino acid in the hemoglobin protein. And so if you recall that people with sickle cell disease have abnormal hemoglobin, it makes their red blood cells kind of a sickle shape. And so what happens, what we found is that individuals that are heterozygous for sickle cell disease. So to have sickle cell disease, they have to be little r, little r. So if they're heterozygous, they're big R, little r. What they found is that they have, their red blood cells, they have some normal cells and some sickle cells. So what does that mean? A normal red blood cell, so that's what it's, and they also have a sickle cell, which is the abnormal. That's a sickle cell. And this has the abnormal hemoglobin, so this is the one that interferes with transfer or transport of oxygen from the lungs to the cells of your body. And so, but what they found is that people who are heterozygotes, they don't have sickle cell disease because they have enough normal red blood cells to deliver enough oxygen to survive. And so, even though they have some sickle cells. And so people with sickle cell disease, they have no normal red blood cells, so they're all this way, which and then obviously homozygotes would have all these. So what they found, and this is why it's called heterozygote advantage, is individuals that are big R, little r, they benefit from protection from another disease called malaria. Malaria, the sickle cell phenotype, prevents the parasite from entering the cells, and therefore they have protection against this disease called malaria. So what happens is that in places where malaria is prevalent, malaria is caused by a parasite. It's a protozoan. It's a one-celled organism, but it's transmitted from person to person through mosquitoes. And so mosquitoes, when they bite the person, can transmit this protozoan into the person, and they develop this disease called malaria. Well, something about having those sickle cells, so being big R, little r, is actually an advantage because it gives them a protection against malaria. The people with little r, that's still not a good thing because the people with little r get sickle cell disease and have trouble transporting the oxygen and so on. But these people with big R, little r, all of a sudden have a better advantage because A, they don't have sickle cell disease, but B, if they get bitten by a mosquito that possibly could give them malaria, they won't die of that. But then the big R people, they're at a slight disadvantage over the big R, little r people because yes, while they don't have sickle cell disease, they don't have a little r, and so there's a possibility that they can contract malaria and possibly die from that. All right, and so therefore, places that are near the equator, near water, have really high incidences of malaria. So that's where you kind of see that. So in those areas, what do you think happened to the sickle cell gene? Did it go up or down in frequency? Up, right? Because now it has an advantage for those particular people in that area.
My grandfather, who's like 100% Irish, wanted to get blood one day, and they tested his blood beforehand and found out that he had sickle cell trait, which is really unusual, especially for someone who's like Irish and has only Irish lineage, that it must have just been a random mutation either in his genes or in like his father and mother's genes that I guess proliferated to him. But it was just really weird because that's something you never see or like hear about or.
So did they not let him give blood? Did they let him give
yeah, no, he can't give blood because of that. Even though he's he's got fine blood, it's just big R little r, so he's got no anemia. But at the same time, if you have someone who also has that, like there's a possibility of getting.
Yes. The safe side. Interesting. And so heterozygotes do not have that full-blown disease due to that dominant normal allele. And so, but they do have that selective advantage. And so that's called the heterozygote advantage, which was really a big deal when they figured this out, and so on. So a mutation that at first glance seems really bad, the little r mutation to have sickle cell disease is not a good disease to have, but it ended up to have some kind of benefit as well. All right, and so this picture just shows you. Here we have Africa here, so there's Europe up here, and so on. And so the dots here is the distribution of malaria, and then it gives you the protozoan name here. So that's kind of showing high incidence of malaria. And the range of colors here is the frequency of the sickle cell allele. And so you can see in a lot of areas that are in high frequency of malaria, there's high frequency of sickle cell allele as well. Okay? All right. So now we have some background about frequencies and so on, and so we're gonna kind of go into some mathematical application of this. So Hardy-Weinberg will only occur if, and this is a review from yesterday. So what I'd like you to do is write those 5 things. See if you can fill in those blanks from yesterday about those 5 things. The first one, the population needs to be extremely large. The second and the third one, it's kind of hard to figure out which one should be which, but one should be that there's no migration and one should be no mutation. It's okay if you don't have it in the right that exact order. Then the next one, mating occurs randomly. And last one, natural selection is not taking place. All right, so all those 5 things have to be true for the frequencies to stay the same. And remember that a population in Hardy-Weinberg equilibrium, that all this is taking place and that the big R's and little r's stay the same throughout the generations, is a hypothetical population, because in real life, usually those 5 things are happening in the wild. All right, so we're gonna relate this to some mathematical equations. The equation used to compare expected frequencies of alleles if the population is in the Hardy-Weinberg equilibrium from the actual frequencies of alleles if the population is not in equilibrium It's called what we call the Weinberg theorem. So what we can do is say, okay, we have this population. If it's in Weinberg equilibrium, that means these 5 things are true and the frequencies will stay the same. And then we can look at an actual population and see, okay, is the frequency staying the same and what's going on that's causing them to change if they are. And so we can see if they're in changing over time. So, for example, let's consider a gene that exists in 2 allelic forms in a population. What does that mean? That's like this example trait I had up here yesterday. So there's 2 alleles for whatever trait this is. There's the big R and then there's the little r. So everybody is either going to be big R, little r, or little r, little r. So, so mathematically, when we figure out the dominant allele and its frequency, we can say the dominant allele has a frequency of P. What does that mean? Yesterday together, we figured out the frequency of the dominant allele in this hypothetical population here. We figured out it was 0.4. We would say that 0.4 is P. P is the frequency of the dominant allele. And then the frequency of the recessive allele is otherwise known as Q. So instead of writing just like the big R like I did here, we're Q. All right, and so that's what those mean from here on out. So then, when you look at a population for a particular trait, all the alleles should either be a big R or a little r. So therefore, these 2 frequencies should always equal to 1, because that's 100% of the alleles. And so therefore, our equation we can do here, if we know the frequency of one of the alleles, we can calculate the frequency of the other allele, because we know that the frequency of both of them should equal to 1. So we have this equation, P plus Q equals 1. So then we can figure out, if we know 1, if we know P, we can figure out what Q is, because we can just subtract it from 1. And then we can do the same thing for P. So we can figure that out here. All right. So before we fill in the rest of the blanks, I'm going to go through a couple of examples. You have some room below on your notes here. I'm going to use the document camera. I'm going to go through a couple of examples with you. I'm going to change gears here. Okay. So example here. We're going to use an example of flowers. So, and I'm going to go through 2 examples here. So example number 1. Oh, let me get rid of this. Get rid of this. There we go. Example 1. So example number 1, we have a population of flowers. And let's say we're going to look at the color, flower color trait. So I'll use A. So red is dominant and white is recessive. So little a is white. And let's say in this population we have a total of 500 plants. And let's break that down based upon phenotype or genotype. Let's do 20. Let's say 20 out of those 500 are white. If 20 of them are white, what's their genotype? Little a, right? And then let's break down the red based upon heterozygous and homozygous. So let's say 160 of them are red, so they're big A, little a. So they're heterozygous red, which means then the rest of the population has to be, which is 320, have to be red and be big A, big A. And if we add these all together, that should equal 500 plants. So that's our population, the makeup of our population. So what we've talked about so far is the allele frequencies. So let's write over here, let's do the allele frequencies. So it's the information about the population. Our allele frequency, we have a total of 500 plants. So how many total alleles for this trait do we have in the population? Each plant has 2, so we have 1,000 total alleles. And so, so now, based upon what we talked about allele, and I'll leave some room for calculation, and then the little a allele. What I'd like you to do right now is calculate the frequency of the big A and the little a allele based on what we talked about yesterday. And given the information that you're given. Frequency of big A, little a allele. Calculator might be helpful. We're going to do a couple of things of dealing with math, so. Alright, so to figure out big A, you have 320 flowers that have two big A's. So you take 320 and multiply it by 2. So you have 640 big A alleles just due to these individuals. But then you also have the 160 heterozygous that each have one, and so that's a total of 160. So then you add these together and you get 800 total big A alleles. And then to get the frequency, you divide it by the total alleles in the population, which would be 1,000 alleles. So you get a frequency of 0.8. And 0.8 here, that is our P. So that's P in our equation. So then, to do little a, we do the same kind of thing. So you have 20 individuals that are little a, little a. So we have 20, and then each of those have 2, so there's 40 alleles with those guys. And then the heterozygotes, the 160 heterozygotes, each have one allele. And so we get 200 divided by that total of 1,000. And so you get the frequency of the little a allele is 0.2, that is Q. And so therefore, hence our equation P plus Q equals 1. 0.8 plus 0.2 equals 1. That's our first equation figuring out the allele. So all the alleles are either a big A or a little a. So building on this, I'm going to teach you about genotype frequencies. So I'm going to go over here and talk about how we figure out the frequency of genotypes. So we have big A and we have little a. So in the population, so people, or the flowers in this case, could be big A, or they could be big A, little a. But when we figure out the frequency of big A, little a, we have to calculate it in 2 different ways, because there's 2 different ways that a person, or a flower in this case, could inherit the big A, little a. They could get the big A from mom and the little a from dad, or vice versa, the little a from mom and the big A from dad. So I'm going to write it in 2 different ways like that, because there's 2 different ways to inherit it. And then lastly, they could be little a, little. Now, how do we tell what the frequency is of these? So we use what's called the rule of multiplication. So remember this, we use this with genetics. So if you remember, the rule of multiplication says that if you want to figure out the chance that 2 things occur together, you take the individual probabilities and you multiply them together, and that's the chance that they're going to occur together. So we want to figure out what's the chance of big A in this population. Well, what we know is that 80% of the genes in the whole population are big A, and 20% of the genes are little a. So if you want to figure out the probability that a big A is going to come together with another big A, you take their individual probabilities. So an individual for big A is 0.8, all right, because there's 80% of the genes. Another big A to come join with it, you have an 80% chance that that's going to come in, so that's 0.8. So we take 0.8 times 0.8, and we get 0.64. So does that make sense? All right, so therefore, on this, we would do 0.8, so big A frequency would be 0.8 multiplied by 0.8, which would give you 0.64. With using the same logic, I'm going to have you go and figure out the rest of the other genotypes. Figuring out, doing these other frequencies based on.
Yeah, heads up, that's definitely instead of.
So for big A, little a, so you have a 0.8 chance and then 0.2, so 0.8 times 0.2 gives you 0.16. Then the second way that's inherited is the 0.2 and then the 0.8 little a from the opposite parent, the big A from the opposite parent. So 0.2 times 0.8 gives you 0.16. So these two collectively, you add those together, we would say that the genotype frequency for being heterozygous is 0.32 because you have to add together both of those. And then little a would be 0.2 times 0.2, which would give you 0.04. So if we take these numbers and add these all together, what do they equal? 1. And that makes sense because 100% of the flowers are going to be big A, or big A, or little a. All right. So therefore, they have to be the same. So your frequency of these three together equals 1. So what I want to do next is talk about. I'm going to have you using the equations here, using P and Q, and thinking about how we came up with and figured out the genotype frequencies for big A. Instead of writing like big A, the frequency of big A plus the frequency of big A plus the little a equals 1, what I'd like you to do is to write the same equation, but instead of writing big A, use P's and Q's. So using P's and Q's, mathematically, how could we come up with an equation like we did here, P plus Q equals 1? What would the equation look like to give us the frequency of big A, little a, and little a? So what I would do is take each one of these individually and think about what you did to figure out that frequency of big A, and substitute in these. All right? And then whenever there's a little a, that's a Q. Yeah, but use P's and Q's, like use the actual letters, P's and Q's.
With P plus PQ.
So if you, let me show you here. So if you look at A, frequency of the big A, that's, another way to say that is P. So what do we do to figure out the frequency of the big A? We took 0.8 times 0.8, which is P times P. We can condense it to P squared. P times P is the same as P squared. So So then, for the heterozygous, we took 0.8, which is P, times 0.2, which is Q, and so that is PQ. But then we had to do it again, Q times P, all right? So that's the same as PQ. So therefore, we can shorthand that by saying 2PQ. Because it's multiplying it by twice, 2 times, because it can happen in 2 different ways. And then little a, Q times Q, that's Q squared. So, and how we write this then, in our second Hardy-Weinberg equation, dealing with genotypes, is P squared plus 2PQ plus Q squared equals 1, all right? And so that's that. All right, so this is the second one. So this one is dealing with genotypes. This one is dealing with the actual alleles. Notice though that you need to know what P and Q is to do anything with this. All right. So let me go through another example that's more realistic. Because real life, can we tell the difference between the big A, little a, and the little a, or the big A flowers? No, in real life you can't. I gave you that to kind of show. Kind of introduce it. So let me give you a statistic about the human population. So let's do another example. Example 2. I'm going to give you a stat. 1 out of 10,000 babies are born with a disease called PKU. It stands for phenylketonuria. It's a recessive disease where they have trouble metabolizing the amino acid phenylalanine. And so therefore they have some issues with it, so they have to restrict that from their diet. So this is a recessive disease. So this is a real statistic. And what I want to do is, from this one statistic, I want us to be able to tell what % of our population is heterozygous for this trait. So how do I go from going from this one statistic to figuring out what % of the population is heterozygous? So I'm going to show you here. So this statistic, we go back up here. 1 out of 10,000 babies are born with this recessive disease. So what's their genotype? Little r, or whatever letter. So these babies, so 1 out of 10,000 babies are born little r for this trait. So my next question is to you, what piece of information did I just give to you in our Hardy-Weinberg equations? Did I give you P? Did I give you Q? Did I give you P squared, 2PQ, or Q squared? I just gave you one of those in this. I gave you, so this is, what is this? This 1 in 10,000, that's the frequency, that's a frequency of people who are little r. So that's a genotype frequency, but it's the recessive one. So what I just gave you is Q squared, all right? So Q squared is 1 out of 10,000. I'm gonna put that in decimal format, so it's 0.0001. I did not give you P squared because, no I did not, because if this is a popular mistake, that means that 9,999 out of those 10,000 have the dominant trait. This is not P squared. Why? Because it also includes the 2PQ, the heterozygous individuals. So you always want to start out with the recessive individuals because those genotypes you know for sure. So if I know Q squared, can I figure out Q? Mathematically, how do you get Q from Q squared? Take the square root. So Q is going to be the square root of 0.0001, which then is 0.01 if you do that. And if I know Q, can I figure out P? P plus Q equals 1, right? So if I know Q is 0.01, P has to be 0.99 so that these guys equal to 1. If I know P and Q, can I figure out what % of the population is heterozygous? Yes. What part of this equation am I really concerned about right now? Just the 2pq. So that's all I need to use is 2pq. I know the frequency of p is 0.99. I know the frequency of q is 0.01. So if I multiply these together, and do that in the calculator, you get 0.0198. That's the frequency of the heterozygous. It's asking you for the percentage, right? So what would I need to do to the frequency? Multiply by 100, and I get it as 1.98% heterozygous.
Just a chance.
It's, yeah, you can't count on that, yeah. You can't count on that. All right, and so what I'm gonna have you do is, if you look at the squirrel packet, little pictures of all the squirrels. This is some Hardy-Weinberg problems I'm gonna have you do for homework. All right. So the squirrel, you have to read the directions. It's pretty self-explanatory, except I hole punched a squirrel out, and so I'm gonna have you draw in a little squirrel that's shaded in. Anywhere on the sheet. It doesn't matter where. Just says draw in a squirrel that's shaded in. Otherwise, you won't come up with the right thing. Just draw in a shaded in squirrel somewhere. That's part of the population. He tried to escape.
Just one of them?
Just one shaded in squirrel you're adding to all the other squirrels on there. So this first sheet is more like the first problem that we did in the class. And then if you turn it over or just look at the next page, there's some problems that are more like that second problem that we did in class, all right, with the PKU. And so, I'm going to have you try those. Let me point out, I pointed out once, but let me point out a couple of things that people always make every year, the same types of mistakes. So the first mistake is kind of like what we just talked about. If I said that 8 out of 10 individuals show the dominant trait, I have every year people wanting to say that that's p squared and take the square root of that. That's not p squared because you include in there the heterozygous individuals. But by giving you this statistic, what indirectly did I give you? I gave you indirectly the recessive, because if you know 8 out of 10 show the dominant trait, then you know that 2 out of 10 show the recessive, and that is q squared. And so then you can, from this, figure out q, then p, and so on. Always want to look at what information am I given that can get me to q, all right? Because you always want to start with the recessive, because those are the only individuals you know for sure. Another common mistake is if it's asking you, if they say the frequency of the recessive allele is, let's say, 3 out of 10, the frequency of the recessive allele, versus 3 out of 10 people show the recessive trait. What's the difference between this wording? Nathan?
For the 3 out of 10 recessive trait, that's 3 out of 10 people who like have the homozygous for recessive trait. And the other one is just like that's how common all of the phenotypes, the little r.
This is the little r. So this is q, this is q squared. So you have to pay attention to the wording, all right? With that. All right, the other, real quickly, the other two things that you picked up, I just want to address before you leave. I know it's time to leave. This one here, there is a small possibility that I may not be here tomorrow. My son is sick today. My parents are watching him. They're leaving for Chicago tomorrow, and my husband has to call in for possible jury duty tomorrow. So anyway, so it's a perfect storm where I may not be here. So if I'm not, this is what this is. If I'm not, we're doing a video and we're gonna start 24. If I am, we're doing a really cool activity to, we're gonna be a little mating population and cause certain events to happen and see what happens to P and Q over time. If I am here, that's what we'll do tomorrow. If I'm not here, then we'll do it on Monday. All right? All right, so we'll just flip-flop these. Now you can leave.